As we start to talk about the regulation of gene expression in eukaryotic organisms, there are going to be some big differences between this and what we had in prokaryotic cells. And the big differences primarily come from the fact that the cells themselves have a different organization to them. And so there is some separation of processes in eukaryotic cells, which will allow eukaryotic cells to have more levels to their actual regulation of gene expression. So if we talk about the first level, or what we kind of think of a lot of times as the first line of defense in this regulation process, this is going to be epigenetic control. And epigenetic control itself is really going to regulate access to the DNA. And so what we're talking about here is that if particular machinery inside the cell cannot actually get to the DNA, then certainly we're not going to be able to transcribe certain genes. And so recall with me that the eukaryotic DNA is going to be organized around proteins. And so ultimately what we get is called chromatin. So chromatin is the DNA organized with a series of proteins, and this is going to be histones specifically. And those histones will be organized into larger structures that we call nucleosomes. Okay, so if we take a look at this, here we can see histone proteins themselves, and they're organized into this larger structure, which is right here, shown in blue. That's going to be the nucleosome. And each one of these individual proteins that makes it up is going to be the histones. So notice that the DNA shown in red is actually wrapped around those. And so the level of packaging around these histones and nucleosomes is going to actually regulate whether or not we get transcription of this gene or not. And so this would be the epigenetic control part of it. So if we have the DNA packaged tighter, we would have less transcription of that area. And if we had a looser packaging of the DNA, then we're going to get more transcription in that particular area. So overall, we have two different types of epigenetic control. We have methylation of the DNA, and then we have acetylation of the histones. And both of these are adding additional chemical groups to either the DNA or the histones. So if we talk first about the methylation part of it, a methyl group is a CH3 group. Okay, so that's just what we call methyl. And so what's happening with the methylation of the DNA is that there are these groups that are actually added right on top of the DNA itself. And what this is going to do is this is going to result in tighter packing of the DNA itself. And so if this is tighter packing of the DNA, we're going to end up with a region that is accessed less. The other option that we have is acetylation of histones. And an acetyl group is going to be this structure. So with this is something that actually gets added right onto the histones themselves. So these would be special little groups that are added on here. And what this does when these are added is this is going to result in looser packaging or looser packing of the DNA. And so the end result of this is that the region is going to be accessed more. So to sum it up, if we did here, in this case, with methylation, we would get less expression, if we want to use that term. And when we're talking about acetylation of the histones, we're going to get more expression. And so we'll find that different regions of the DNA will have differing amounts of methylation of the DNA, and they will also have different amounts of acetylation of the histones. And this is something that's going to be fluctuating really all of the time to tightly and carefully control the level of epigenetic regulation of the DNA.